Good, thank you. This is some experiments we did with SVE, and not in real life. We had the Gem5 simulator, and there's an extension to it now that allows you to run or simulate, cycle accurately, SVE workloads. So we picked this. It's not supposed to be an advance in genomic sequencing. It's just a sort of dense matrix or dense vector kind of application. So I'll go through it quickly. I've got quite a few slides. So we were looking at pick an emerging application and then experiment with this new accelerator that ARM have added to their instruction set. And we did 3 versions of it, of the basic genomic matching, and there have been improvements and so on, so I'm not going to go into those. This wasn't an algorithm study. The basic Smith-Waterman algorithm, and 3 ways of doing it. One is to batch it with separate lanes of the vector machine doing separate sequencing matching, but obviously in lockstep. Slice, which is, all these algorithms are fairly well known. Slice just means that we speculate when we try and do the match. I'll say more in a minute. And then a method that's sort of a wavefront sweep through the score matrix on the Smith-Waterman. It gives you some, all of these things have certain degrees of parallelism. So here's something many of you have probably seen before. It's just the story about genomics and computational biology and how quickly they're quite proud of the fact that they beat Moore's law. My view is they came off of such a low base they couldn't help. But anyway, they've accelerated. If you look at the top there, you've got some idea of why you can do these ancestry things where you spit in a thing and they'll give you a measurement or tell you about your something about your genomes, maybe. So this stuff is getting much less expensive, and of course, one thing that we'd like to do is speed the process up, make it cheaper. Now, there's lots of components to this. I'm not suggesting that the only thing is just to speed up the computation. In fact, the secret sauce is usually in the transduction of creating these fragments, but here's what we're trying to do roughly for people that are not familiar with this, and you're certainly not listening to an expert on the subject. So you've typically got a reference gene, and what you've got is these splices that you get from the samples of people's genomes. They're split up into base pair sequences. So you can just think of it as a sequence of 4 different letters, A, G, C, and T. You're just trying to match a snippet against the reference gene. And with the possibility that there are some letters missing and some letters added in the sequence. So you can't get a perfect match. What you're trying to do is get the best match. And then you can assemble these pieces. I've tried to show there in, see if this does the job. No, it's just not strong enough. Okay. It isn't going to work for me now. Do we have a better one that I can take home with me? Oh. Okay, I'll point.
Try that one.
This one.
Yes.
Thank you. Yeah. Now, I was planning to point it with your eyes, actually, but yeah. So, okay, we're just trying to assemble these into the best match to that reference. I think you've got the idea. So getting this is, an interesting technology. What we're doing, of course, is just the computational part. And if you look at these steps, the big step is you've got extraction, of course, sequence alignment, assembly, and analysis, which comes later. That's when you decide what the person or what the gene says, you know, it's what's different, what's wrong, what's right, and so on. This is the part that computationally takes the time. It's just the matching of those two sequences of 4 letters, to put it in very simple terms. And this is the thing that the ARM SVE that we were playing around with, and what we had was the opportunity to have a variety of vector lengths from 128 up to 1024, and per lane predication. So that was a nice feature for some of the algorithms, it turns out. And it also supports a gather, load, scatter, store mechanism. And one of the things that we're doing is to see if there's any way we can improve. Is there any things we could change in the architecture to make it better, or in the microarchitecture? And because we originally authored Gem 5 at Michigan, it's, I wouldn't say easy, but we can actually go in and play around with it and essentially change the microarchitecture. Obviously not the software interface. That's much more. We can change it, but then of course we're stuck with no software. These are just 2 offerings out there that give you these sequences. So think of the base pairs as the letters, or pairs of letters actually. And you could get your snippets come, you know, in 100 to 900, sort of that range. And depending on which machine you buy, and I'm sure there are other offerings out there now too. The nice thing about SVE is it's vector agnostic. So you can use the same code, the same assembly code for varying vector widths. So we explored, it made it easy for us. We didn't have to keep rewriting everything. So here's our target algorithm. Let's just go over the algorithm very, very quickly, and I'll try and abstract it away from the actual gene, you know, the bioscience. Here's our reference sequence. We want to see where this query sequence best matches in it. Remember, this is going to be from, say, 30 to 900, and more modern machines tend to give bigger snippets, which makes life easier. We pick up a lot of these things and we figure out how we can assemble them to line up against the reference finally. And the Smith-Waterman algorithms are sort of an ancient algorithm, but it's provably optimal. And of course, there have been improvements on it that are not necessarily optimal, but much faster. But we just stuck with this. And it works, it's a sort of dynamic programming technique that works with a scoring matrix. Let me just show you what that means. The output is the match. I just think I just said that to everyone. So here's our matrix that we're scoring. We've got a reference sequence, and you can see some letters along there. So that there's 4 different kinds, A, G, C, T. And this is the query sequence, and we want to know how best does this sequence start with the A there, and so, or the here, or whatever, okay? So the scoring method is these set of interrelated or dependent functions, and H is the final number that goes into the square. So you sweep it, and you figure out H from the point is, if you look at the indices in these things, you'll notice that the value of something depends on the value of things that are in the northwest corner, north, northwest, and north, and just west, so that you've got this sort of dependency going diagonally down this part. So you calculate these from older values of H and previous values of E and F in this case. These are constants that you plug into the system, similarly here. And then the interesting thing to note about these functions is they're nonlinear, they're maximums. So sometimes there isn't a dependency, because if this is not the maximum, this thing doesn't depend on the previous version. I'll try and explain that a little better in a moment. But we're going to sweep through this thing. So I've figured out this is the scoring matrix. Let's just focus on this. You can see that the dependence on getting this number comes from above, which is fine. That means you can certainly calculate all of this row as far as the green part's concerned. Not so good with the what's across the row, because the dependency runs across from left to right. And then you've got this diagonal dependency from the top left corner here to get the final H, and then you put these together and you max all of these things. Now, if it turns out that this thing doesn't occur in the max, you've broken dependencies crossways, so you can speculatively parallelize the algorithm. That's really the point I'm trying to make. So if I fill a thing in, once you've got that, you backtrack along the maximum steps, and what that tells you is this matches here, A matches there, C matches there, and then there's an insertion, because you've made a vertical move. If you made a horizontal move, that would correspond to a deletion, and so there's an insertion. Indeed, there is a G. So if you allow that insertion, the thing's not perfect match, but it's the best match, according at least to this, this matrix that we've calculated. So this is our backtracking. Backtracking's not a big deal. It looks sequential. A little bit of thought tells you can actually keep the partial backtrack values while you're doing the forward sweep to build the matrix in the first place. But it doesn't matter, because this just doesn't take much time anyway. So, and this is what I said, the path direction, if it's diagonal, it's a match. If the path goes up, like it does here, there's an insertion, and if it goes across, there is an example here that means there's a deletion in the query sequence with respect to the reference sequence. So that's our match in this little example. So let's look at the different things that we tried on SVE. We tried a batch system, a slice, and a wavefront. And these are our descriptive terms. They're well-studied algorithms in the literature for the most part. We're not inventing algorithms here much. There are some differences. The batch thing, we just send the sequences down the lanes, down separate lanes. Of course, they run the same code, but you've got the reference sequence may be the same one in each lane, but then you send different snippets down. So that's how you get your parallelism. And in this case, you'd have the vector width. The number of lanes would be the number of things that you're trying to match, the number of snippets, if you like, that you're trying to match, query sequences that you're trying to match simultaneously. And in the slice, what we do is we take that matrix and we just work on one match, one query sequence, one reference sequence, and we break it up across, we just go across the row and break the dependency. I'll show you in a minute, and then maybe we'll come and fix it up later. But what that lets us do is to use all the lanes concurrently. But there may be a fix-up. And then wavefront, you remember in that scoring matrix, the dependency was from the west, north, and northwest. So if you calculate things along a wavefront like this, we just called it a wavefront, you can get your parallelism. You've got to play around with the indices a little bit carefully at first, and that's where predication comes in handy to program something like this, because you've got to start up essentially when this diagonal is not as wide as the number of lanes, and then once it gets going, you can start flying. So we looked at these three things. So our purpose wasn't really to show that this is good for speeding up genomic matching. It turns out it is. It's pretty useful. More, it was to take some reasonably realistic application and see what things we felt were missing from the microarchitecture. So this is the batch. They're all going down, separate queries going down separate lanes of the vector machine. They all try to match against the same reference sequence. So I think you can get the idea. This is kind of simple, it's kind of multi-threading, except, well, the threads are in SIMD threads now, not Unix threads. Anyway, maybe too much animation. So here's the slice. As I said, we just calculate across here. And remember, this function is a max function. So if this is not bigger than these two things, you can do all these things independently. If there is a case, you may have to go back and fix it up across these boundaries. So each lane is going to do this slice, and the next lane that slice, and the next lane that slice. And then maybe we might have to adjust those. It turns out not as much as you'd think. It's not a bad approximation, and the fix-up stage doesn't cost you much. So at first it seems bad. It's all down to the fact that in this case, it's not often the case that this is the biggest term in that list of three. Without looking at all the indices or anything, that's sort of the takeaway. And because of that, we're just dependent on these things, which we've already got up on the row above. Okay, so yeah, we've seen this before. So if that F function, so here it is. I'm trying to diagram now the fix-up for the sliced approach. Occasionally, you're speculating that you don't need to know about this in order to start this sequence. And when you do, you just go through and adjust all those values by adding a value to them. The point is that the red arrows don't happen very often. Interesting fact. Well, at least on the samples we looked at. So I shouldn't say that as if it were a law of nature. So this is the slice, and here's the. I'm just trying to diagram the fix-up in yellow here. Sure, I'm not running out of time here. Okay. So the wavefront method is as I diagrammed, you just use these dependencies, these functions that you're taking the max of here to get the blue box, and sweep through here. So you completely match, this is completely data flow wavefront, if you like. And the trick in terms of, let's see, we've got a better diagram. Yeah, you can see the thing going through here. The point is that, as I mentioned before, this grows and shrinks as you go through the matrix. So these things are going to be on the order of hundreds. The reference is going to be on the order of billions, actually. So this thing is going to be slowly growing. So when you first fill up the pipeline, it's not going to be fully utilized. But you can write the same code for the whole thing because you can use the predication feature of the SVE. Okay, so here it is. Enough of that. There's more bookkeeping in the other ones, but as I said, it's helped by predication. So several things that were nice about SVE. One was it's width agnostic, and also the predication makes easier for the code writer. You don't have to have prologue and epilogues that are different in your loops. Here's the setup. So since we have got control of the simulator, we can compare it to just a single core ARM, and here's the spec. It's a V8 architecture, nominally running at a gigahertz. We just assume clock ticks our gigahertz rate here to do this. It's an 8 issue, so it's a pretty fancy machine that we just set up. And then then the SIMD width was either a 128-bit neon or any one of these 4 SVEs. And we sort of used the single core as the baseline, compared it to the neon, and then compared those to these 4 vector widths. And here's some cache sizes and so on. Now, the point with this is, and the DRAM's a very simple model. Okay, it doesn't model a DDR interface. We have done that, but that's work in progress. So you take these results with a grain of salt in the sense that this is a very, very simple DRAM model. So it may change the relative merits of the different algorithms. That is something we haven't dug into in great depth. Also, the caches, we haven't swept through different cache sizes. And we have the option to do all those things, but this work is something that's just been done in the last month or 2 by one of my graduate students. And here's the setup, 3 different versions of the algorithm. They're all exact versions. The slice, even though it's speculative, fixes it up in the end correctly. A reference was from an E. coli gene, which is a small gene. I think the human genome is about 1 billion or so base pairs, so this is quite small. So our reference was relatively small, and these are the queries. We had about 1,000 of those. And we looked at it up to about 400 base pairs. And so let's take a look at some results. This is the speed up over the CPU, these numbers. So the little blue thing, which is that 8-issue ARM CPU running at 1 gigahertz, if there is such a beast, is 1 on all of these. And you can, then we looked at the thing running on the neon, so these 2 things here are batch neon and slice neon. We didn't do the wavefront on the neon, but you can see there the next 2 figures. And the number on top is how much faster are they than that 8-issue core. Now, these all run, the neon and the SVE also run at 1 gigahertz. The next group of things are using the SVE. So we've got batch, sliced, and wavefront, and you can see that the sliced actually gives pretty good performance, particularly as the query sequence gets bigger, which my understanding is modern transducers that actually sample and create these sequences are moving in the direction of longer and longer sequences. So they're capable of dealing with the fragility, I guess, of those sequences. So the speed up's quite dramatic, actually. And this is for, let me see, so we're using 16-bit vectors. Wavefront uses 64-bit vectors, which puts it at a big disadvantage, actually. Okay, yes, the number is there. We're just looking at the 128-bit vector width. So the wavefront actually only has 2 lanes in it. So in a sense, this is a bit of an unfair comparison to the wavefront. There's lots of other experiments we need to do to clean this thing up. So I'm just throwing out the initial results, which are promising. And I don't want to claim that you need to use this beast to do your genomics. Although actually I think you probably do, but this is not conclusive, but interesting. Okay, now let's look at, we've got different sequence lengths, and we're taking the number of instructions that are executed. And you would expect on the vector machine there to be far fewer, because basically your executing a lane's worth of stuff in one instruction. And indeed, that's the case. Just looking here, this end of this thing is where we're using the vector machine. So we've got the basic machine, which is 1. Then we've got the neon cases, which are the orange and gray there, just the different implementations of Smith-Waterman. And it looks like that the batch seems to work better on the neon. And then we've got these 3 on the right-hand side, which are the sliced neon, sliced, I'm sorry, not the sliced neon, yeah, that's right. That's right, there's actually, this has been switched around on me. So batch SVE is the gray one, which seems to be doing better than its equivalent neon. So now, these have been grouped so that we're comparing the same algorithm, or say, on a neon and a SVE, and one is our 8-issue out-of-order ARM core. And I think what you can see here is that let's see what the best things are, is the sliced SVE with 128-bit vector seems to be the best performer. Wavefront's not bad, but it's hampered by the fact that we were using the 64-bit data path in the lane. So the speedup's quite interesting. And here's some figures of control and the bandwidth to memory as the sequence length changes. So in batch, it just goes up as you'd expect. You're just doing more work. But it actually goes down in some of these because you don't need to you reuse data in these cases. Okay. Now, the difficulty with these figures meaning anything is that the memory model is kind of very simplistic. Okay, so let's look now at speed up of the waveform algorithm with different sequence lengths. You can see the speed up over a CPU, and what we've done here is we've compared. So one of the criticisms we had is, hey, you didn't do the CPU in assembly language. I guess what I should have told you, which I didn't, is that we ran regular C version of the algorithm on the single core and then hand-coded everything on the SVE, which was quite tedious. Vector agnostic and predication helps, but it still means you have to do a lot of nitty-gritty assembly language coding. So you can see when we took the CPU, which is our baseline, that when we, CPU, okay, so if you compare this, the gray, with the CPU assembler, you can see that you can get a speed up of 3 or 4 almost just by hand coding the basic algorithm. It's about a 50-line program, the kernel of it. So what we should really be comparing our speedups in the case of sequence lengths of 25, 50, and 100 are these numbers here, 3.8, 5.7, and so forth. And then the 256-bit vector is a big improvement, but it's nothing like the improvement that we were first seeing. So the bars have slightly come down. It's still a big deal. For these longer sequences, it's still about 3 times quicker. And there's a lot of wrinkles in the algorithm that need to be ironed out too. So this is just comparing assembler for the same thing. So that was our readjustment of the results based on apples to apples rather than out of the box C program versus hand coded SVE. And this is just a wavefront algorithm with vector scaling. In general, it gets better as the vectors get wider, which you'd hope. And it works better as the query sequence gets wider. So you're just making it more of a dense vector problem in both directions there. So if we look at vector scaling for different algorithms, we can see that over here we've got batch, slice, and wavefront with a fixed query length of 100. And as we grow, interestingly enough, the slice starts to fall off as you increase the vector size. The wavefront continues to show improved performance as you get more and more, you get a wider vector, basically. And then over here, similar performance. This time, instead of the query sequence being 100, it's 400. So you can see that the graph goes a little, maybe 30% higher. Now, at some point, you reach diminishing returns, and you can see that if you look at sequence lengths versus the 3 different kind of implementations of the Smith-Waterman, you get different regions. Batch does better for the smaller queries, wavefront in the middle, and sliced in the top end. So as you get longer, it seems like you should be using the algorithm. So, conclusions. It seems to work. It seems to do a pretty good job. Speeds things up. The vector system works. Predication was very helpful. Agnostic vector width was helpful in terms of just code productivity. There's much more to be done. The memory controller is very primitive. I've got some results for that. What I can tell you sort of qualitatively, what they do is they change this graph when you actually use a DDR model, a real DDR model of the DRAM instead of the simple one that we used. The slice doesn't look quite as good anymore. Okay, so questions.